Good morning, respected managers, colleagues, and scientists. Today, I'm very excited to get Best Research Award. I'm honored to present our research. Peroxidase from foxtail millet bran exerts anti-colon cancer activity while targeting cell surface GRP78 to inactivate state 3 pathway. I'm Prof Zhuoyu Li from China, Shanxi University. The global cancer statistics in 2020 showed that the incidence of CRC ranked third in all cancers and second in mortality. There is a recent trend year by year. CRC is a major public health problem in nowadays. For CRC treatment, targeted therapy has become an effective way. Some plant proteins have anti-tumor activity with strong specificity and low toxicity. They may play important pharmacology effects. Those proteins include three major types. 1. Plant lectin. 2. Ribosome inactivating proteins. 3. Serial proteins inhibitor. The millet bran is rich in protein up to 18 to 22%. Traditional Chinese herbs were suggested proteins in millet have good nutrition for intestine and stomach. Our previous study found that the peroxidase from foxtail millet bran, a 35 kDa protein, can inhibit cell proliferation, migration, and remodel tumor metabolism. It indicates that FMBP has a significant anti-CRC effect. However, the metabolic pathways by which it exerts anti-CRC effect are unclear. Our previous studies showed that FMBP exerts anti-colon cancer effect through increased dose. The result indicated that FMBP didn't enter cells, mainly located on the surface of CRC cell membrane. Therefore, we wonder, what is FMBP targets against CRC? By pull-down and silver staining, a different band was screened. The band was identified by mass spectrometry as a ER molecular chaperoning BIP, GRP78. Biolayer interferometry assay showed that a direct interaction between FMBP and GRP78. Next, Figure E showed that GRP78 is still stable at 80 degrees when interacted with FMBP. It indicates FMBP efficiently protects cell surface GRP78 protein from temperature-dependent degradation. Further, the interaction was verified by computation inhibition assay in Figure F. It has been known that glucose-regulate protein GRP78 is a major chaperone in the endoplasmic reticulum of normal cells, while in tumor cells, GRP78 is discovered in multicellular locations. Particularly, GRP78 abnormally locates on surface of cancer cells, but not in normal cells' surface. Therefore, GRP78 can be regarded as a molecular target for tumor-specific therapy. The immunofluorescence assay confirmed that FMBP and cell surface GRP78 exhibited co-localization on the cell surface of CRC. As presented in Figure 2B and C, after FMBP treatment, DLD1, HCT116, and HT29 cells with a high expression of cell surface GRP78 saw a lower cell survival rate compared to LS174T and FHC cells with a low expression of cell surface GRP78. It suggested that the anti-CRC effect of FMBP is positively correlated with the expression of cell surface GRP78. Furthermore, in Figure 3A, B, C, blocking cell surface GRP78 with anti-GRP78 antibody reversed the inhibitory effect on cell proliferation and colony ability induced by FMBP. In Figure 3D, E, F, G, after blocking cell surface GRP78, the pro-apoptotic effect and the growth level induced by FMBP also showed significant reduction. These results indicated that blocking cell surface GRP78 significantly impairs the anti-CRC activities of FMBP. Studies reported that trial treatment can induce the translocation of GRP78 from the ER to the cell surface. The results indicate a significantly increase in cell surface GRP78 upon treatment with trial. As expected, the effect of FMBP on cell survival was significantly reduced, and the apoptosis rate was significantly increased. This indicated that the anti-tumor effect of FMBP was significantly enhanced. LS174T cells expressed lower GRP78 within overexpressed GRP78 in LS174T and detect the effect of FMBP on cell survival rate and apoptosis, which was consistent with the result of trial induction. In addition, in overexpressed GRP78 LS174T cell, the induced effect of growth accumulation by FMBP was further increased. These results indicate that GRP78 is a target of FMBP against CRC. GRP78 is composed of two domains. One is the nucleotide binding domain, NBD, from valine 31 to glycine 407, and the other is the substrate binding domain, SBD. Studies reported that NBD induces conformation changes of its SBD and thus triggers the anti-apoptic activity of GRP78. The NBD of GRP78 is necessary for its anti-apoptic function. Recent studies showed that the residues asparagine 400 to 500 of GRP78 were responsible for translocation of GRP78 from ER to plasma membrane. Hence, the truncated body of GRP78 and 500 was constructed. As expected, cell survival rate was significantly reduced, and the apoptosis rate was increased in GRP78 and 500 overexpressed cells. Similarly, Rho's accumulation induced by FMBP was also enhanced in GRP78 and 500 overexpressed cells. These results suggest that FMBP plays an anti-CRC role by directing binding to the NBD of cell surface GRP78 and interfering with the anti-apoptic activity of cell surface GRP78. Next, we investigate the downstream factors of cell surface GRP78. We previously found that FMBP can inhibit STAT3 phosphorylation and increase intracellular growth. In addition, research shows that STAT3 and growth can regulate each other. Interestingly, co-IP assay showed that GRP78 interacts with STAT3 in CRC cells. Furthermore, STAT3 may be a downstream signal factor that FMBP exerts anti-CRC through binding to GRP78. In Figure B, FMBP-induced STAT3 phosphorylation was drastically neutralized by blocking GRP78 in CRC cells. In Figure C, overexpressed GRP78 or GRP78 500 aggravated the reduction of phosphorylase STAT3 and the STAT3 levels. In Figure D, increased dose level by FMBP was abolished by the activator of STAT3. In summary, the interaction between FMBP and the GRP78 reduces phosphorylated STAT3, leading to an increase in growth, which in turn inhibits the expression of STAT3. Next, the xenografted nude mice model was constructed with normal LS174 T cells and GRP78 and 500 overexpressed LS174 T cells, respectively, followed by treatment with FMBP. In Figure B and C, administered with FMBP reduced tumor volumes in GRP78 and 500 overexpressed LS174 T cells, while had little effects in LS174 T cells. The result of immunohistochemistry assay is consistent with the cellular level. That is, only in GRP78 and 500 overexpressed tumor tissue, FMBP reduced the level of phosphorylated STAT3 and K67 and increased the activated caspase-3. In conclusion, our research demonstrated that cell surface GRP78 abnormally expressed on CRC cell surface, but not on normal cell surface, is an underlying target for FMBP against CRC in vitro and in vivo. Our data support FMBP has the potential to be developed as a target drug for the treatment of CRC in the future. The above research has been published in the Active Pharmaceutical Clinical B in October 2021. Thank you for your attention.